Hello, everyone. Welcome to the 7th installment of the High Elevation 5-Needle White Pines 2024-2025 Science and Management webinar series. I'm Enzo Martelli, a PhD student in Karen Nelson's lab at University of Montana, and I'm co-organizing this webinar with Lou Deloise, a PhD student in Danielle Ulrich lab at Montana State University, and Karina Lee, a master's student in Diana Tombach's Forest Ecology lab at CU Denver. We have another great presentation for today, and I'd like to start by introducing our Executive Director, Julie Schamhart, who will share a few announcements with us. Welcome, Julie.
Thanks, Enzo. And thanks, everyone, for being here today. I just had a couple of announcements I wanted to make. And the first one is, you can all see this, but this I have in front of me is the very first issue we ever printed of Nutcracker Notes. And if you want to think for a second of when you think that might have come out, I'll tell you when I'm done talking here when this came out, but it has a little guy on the top. This is supposed to be a nutcracker, I believe, that someone made. I don't know if this was on a typewriter, but it's like parentheses, and it's kind of like the way people used to make emojis. So it was a very long time ago. We have another, it came out on April 1st, and we have our spring Nutcracker Notes is coming out. Should be in May, into May probably. It is right now they're doing layout on that. So anyone who is a member of the White Bark Pine Ecosystem Foundation will be receiving our next issue of Nutcracker Notes. So yeah, we would like to, if you want to renew your membership, you get that issue. Anybody can read the older issues in Nutcracker Notes, and I'll put a link in the chat box here in a few minutes where you can find those older issues of Nutcracker Notes as well. So we have that coming out, and then we also have an email newsletter coming out at the end of April. And Lou Deloise is working with me on that to do our layout on our email newsletter. Everyone can get that. So if you're not on the email newsletter list, get on our website and sign up to join the email newsletter list. It has things like jobs, latest research articles, summaries of articles, anything that's been in the news about high elevation 5-needle pines, events, volunteer opportunities. We just took a group of volunteers to the Coeur d'Alene Nursery just a couple of weeks ago. So if you're interested in opportunities like that, get on our email newsletter list. And like I said, that's open to anyone. So we would love to have you join that. And the last thing, I wanted to just take one moment to thank our federal agency employees for all the work that you do. And Sharon's here today. And if you guys want to take a second, and anybody wants to react with a clapping for all of our public agency employees. And yeah, thanks for all your work.
Thanks for joining us today, Julie. And if you miss any of our previous installments, you can find the recordings of these talks on our YouTube channel, White Bark Flakes. The link to our website and that YouTube channel as well will be posted in the chat promptly. Finally, after the presentation, we will have a Q&A session where you can share all your comments, doubts, and questions. So please post them on the chat box, and our team will be collecting them. Now, it is my pleasure to introduce today's speaker, Sharon Hood. Sharon is a research ecologist in the U.S. Forest Service Rocky Mountain Research Station Fire Science Lab here in Missoula, Montana. Sharon earned her PhD from the Organismal Biology and Ecology program in the Division of Biological Science at the University of Montana in 2014. And since then, she's focused her research on understanding fire-induced tree mortality, pine defenses, and the ecological effects of treatments in fire-prone forests. And with that, I'll turn it over to Sharon. Welcome, Sharon.
Great. Thank you, Enzo. Thanks for having me. Let's see, start my presentation. Let's see if this goes. All right. Can you see that?
Yep.
Correct. Okay, great. Thank you. All right. Yeah, well, thanks for coming to this webinar today, and I'm going to be talking about some defense traits in high elevation pine species, and just how the kind of short answer to the that I'll have at the end of the talk is that these traits are really inconsistent and species dependent. So probably don't need to tell this audience too much about mountain pine beetle, but it is a native beetle that's capable of attacking and killing healthy pines and can cause high levels of mortality in pines throughout Western North America, including Canada, even though this map only stops at the US border. But this map here just shows the level of mortality in pine forest in the past from 2000 to 2019. And you can see that there's been extensive levels of mortality in many of our forests. But and these beetles are capable of are increasingly causing more mortality at our high elevation pine forest. That in the past, climate was typically too cold for mountain pine beetle to establish very frequently, and to where outbreaks really were very infrequent in our high elevation forest. But as climate warms, outbreaks are becoming more common here. This is a picture from the greater Yellowstone ecosystem and whitebark pine. So all those red trees are dead and dying. Whitebark pine trees. And so there's, as with climate change, there's concern that these beetles will cause increasing pressure on our high elevation pine forest. But our trees aren't just complacent bystanders waiting around for bark beetles to attack them. They have a lot of defenses to try to ward off attacks. I'm going to go into more detail about the defenses in a little bit, but just so you know, pines have are just chock full of constitutive and induced defenses, like this resin here, and there's a defense system throughout the tree, both in the resin and phloem. And so, again, I'll talk about this in more detail in a little bit, just so you can understand some of the variables that I'll report on. So the objective for this study and the talk today is to quantify the constitutive and induced defense traits in 4 high elevation 5-needle pine species, and explore how these defense traits correlate with each other. So we have four pines, and I'll mostly be referring to them by their 4-letter species code, but we have Great Basin bristlecone pine, or Pinus longaeva, Rocky Mountain bristlecone pine, and Pinus aristada, foxtail pine, Pinus balfouriana, and limber pine, Pinus flexilis. So I'll try to use the common name throughout in case you're not familiar with the scientific name. So why these species? Again, mountain pine beetle is causing more and more high levels of mortality in high elevation pine forests due to climate change. And it's real, even in 2025, our range maps are still highly inaccurate in these remote high elevation forests. And is in 2014, when this study began, there was more in the aerial detection surveys that the Forest Service does to document mortality and the cause of mortality was showing, mortality spots were showing up in areas with in bristlecone pine and foxtail pine forested areas. And it was unclear if that mortality was actually especially in Great Basin bristlecone pine, or if it was in the co-occurring Pinus flexilis or limber pine. So, so it was really hard to know like how much mortality was actually occurring, and it was unclear if Pilo and Piba were hosts to mountain pine beetle. So we initiated a study that was led by Barbara Bentz to determine if Pilo and Piba are host and to quantify their constitutive defenses and growth. And we focused on sites that co-occurred with limber pine to try to know that where we knew there was bark beetle pressure in the area, so we could tell or test if mountain pine beetle was there and just not attacking these pine species, or if and which species were being attacked. And another reason for these to use these 4 species is that they're really long-lived species. These all live in 1,000 years or more. Pinus longaeva is the oldest known tree species a single organism tree species, single trunk, compared to like aspen or the clonal species. So these trees, live a really long time, and they've experienced range expansions and contractions with climate, and those range expansions can likely bring them into contact with mountain pine beetle over evolutionary time. So there's reason to think that in the past, these trees have evolved under pressure with mountain pine beetle, but maybe with their the recent range contractions from past colder climates, that there's been less pressure from mountain pine beetle, but they still have many defense traits retained. And so this is just an example of one of the sites in the Ruby Mountains in Nevada. And it's really difficult to tell some of the species using remote sensing tools. So this is sometimes it's a great opportunity to get out in the field and spend some time in the high country. So we started this study in 2014 and focused just on bristlecone pine and foxtail pine and limber pine. And so this study, it was only on constitutive defenses and looking at resin chemistry and resin ducts, and then also just establishing which species were being attacked by mountain pine beetle. In 2017, we added another study. Actually, I should go back for a little bit for a second. And the outcome of this study was to show that Pilo, Pinus longaeva, was rarely attacked by mountain pine beetle, and also foxtail pine was very rarely attacked. They don't seem to be strong hosts, and further research by Barbara Bentz and others have further established that those species have higher constitutive resin terpene levels and resin ducts than Pinus flexilis. So with that knowledge, we wanted to add a second study that we called the 2017 study. And that was then to look at the sister species of with Rocky Mountain bristlecone pine and where it co-occurs with limber pine. In this study, it was we looked at constitutive and induced defenses with growth, and also added phloem duct defenses. So that was a study that David Soderberg led for his PhD, and that we found that there was high inducibility of resin chemistry, especially in Rocky Mountain bristlecone pine or Pinus aristada, but very few trade-offs between constitutive and induced defenses and growth were found. In 2017, we wanted to go back to the 2014 study sites and add induced defenses. So unfortunately, and the hindsight's kind of 2020, but in the first study, we didn't tag the trees. So we went back to the same sites, but we don't have the same trees. But we collected constitutive and induced phloem and but not growth cores in that study. And that study was led by Justin Runyon, and again, found no trade-offs in chemistry, but higher defense levels generally at the coldest and driest sites. So we wanted to combine the information from these three studies to kind of put things together, especially with focusing on resin ducts and how resin ducts relate to resin chemistry. And so this is my very crude map right now that I'll need to improve at some point, but this is, you can see all 12 of our sites. The purple squares are from the 2017 Rocky Mountain pine, bristlecone pine study. And then the other sites are all from the 2014 and 2019 studies. And so briefly, this changes a little bit for each study, but we have our 4 species, we have 12 sites, and we have day 0, which is what we call constitutive defenses. These are baseline defenses, and then measured day 30 defenses for induced defense. The trees were induced 2 different ways. One was through mechanical wounding, and one was through inoculation with Grasmania clavidra, the fungi that the mountain pine beetle is associated with. And that's the simulated mountain pine beetle attack for that induction. And then we collected increment cores for growth and xylem resin ducts, and phloem for phloem resin ducts, and also for resin chemistry at both day 0 and day 30. Okay, so getting back to pine defenses, I kind of want to just pause here and kind of give a little background about defenses so we understand what we know so far about how these traits work together and why we measure what we do. So resin is the actual defense that pines have. It's a complex mixture of terpenes, and this is it acts as a physical and a chemical defense, and that resin flow as the beetle starts boring into the tree. The resin flows out and acts as a physical deterrent that can stop the attack as the beetles get trapped in the resin. And it also many of the terpenes are toxic to bark beetle metabolism. So we know that resin is the actual defense, but the resin duct system in pines is a very important part of defenses, because that's where resin is synthesized, stored, and transported to attack sites. So looking at this picture of the pine wedge, there you can see these kind of lines. I don't know if you can see my my mouse, but so there's horizontal ducts and vertical ducts that are form a network of throughout the xylem of the tree and extend out into the phloem. And all pines are have this resin duct system. The picture that's of the slide with the blue shows the epithelial cells of the resin ducts, where resin is synthesized, and then it's stored in the lumen of the ducts. This is the large duct is the vertical duct, and then it's transported in these horizontal ducts. And you can see they're much larger in size compared to the red stain cells of the xylem tracheids. And so that large size makes it nice for tree ring analysis, which it shows on the bottom picture, where these resin ducts get embedded in the xylem of the tree, so that we can go back in time and see how many resin ducts were being produced in the tree annually, and it makes it relatively simple to measure without a lot of more complex microscopy. Phloem ducts are much less studied, but we decided to focus or add this to our study because, as the phloem is obviously on the outside of the tree and where the bark beetles are spending the majority of where they're feeding and the larvae are developing. And so we wanted to try to quantify the phloem ducts in the phloem, the ducts in the phloem. And that's, so you can see here in this picture of a cross section of the phloem, and you can actually see some of the annual rings that are produced in the phloem itself. So there's lots of, so these pines have lots of defenses, and but we know that resin itself is the main defense, but it's a hard thing to measure. And so we can measure resin flow, but we know it's highly variable, and it's really a kind of a snapshot in time. This is just from one study showing the variability over just one season from 2 different sites. And you can see that resin flow changes monthly. It changes depending on the site. We know that it changes with temperature. So it's just, even though resin again is the main defense, it's hard. It's hard to actually use as the defense trait. But luckily, we know that resin ducts are positively related to resin flow. So here we have this graph showing as total duct area on the x-axis and showing how it's positively related. So you have more area in ducts, and that leads to increased resin flow. And so this resin duct quantification is much more repeatable and consistent. And like I said before, it's also allows us to retrospectively look at how defenses have been changing over time. So it's it's much more detailed than just that snapshot of resin flow that day. So this is a cross-section of an increment core, and there's lots of different ways to measure resin ducts. So I just want to go through them because I'll be reporting them in the results. So here you have on the right side the phloem, and each light and dark band together is 1 year's worth of growth. You can ignore the big pencil mark dot that was used for dating. But the first step of quantifying resin ducts is to actually date the cores to where we have an actual calendar year assigned to each tree ring. So we know the year that ring was formed. And then we go back and measure the individual resin ducts and assign those resin ducts to the calendar year. And so because we measure the size of each duct, we can calculate the average duct size. The number of ducts per year and the total duct area. Those are what we call our unstandardized ducts. And then we can standardize them to growth through duct density and relative duct area. And there's been several studies now that show that trees with larger ducts and more resin duct area are more likely to resist a mountain pine beetle attack. This is data from white bark pine from Kitchis et al. In 2020. Here I'm just showing the 3 resin duct metrics that were most significant in the model of tree resistance. And you can see that the green, those are the trees that survived a mountain pine beetle outbreak, have bigger ducts, more resin duct area, and higher relative duct area compared to the orange trees, which are the ones that died during the outbreak. And again, this has been shown in numerous studies now. This was a meta-analysis by Vasquez Gonzalez of 8 different studies where they found that it was only trees that, only resin duct traits that were related to area or size were related to resistance to beetle attack. So each one of these squares is showing different independent studies, and the diamond is the overall mean, and the resin duct area or size is the only one that doesn't overlap with the 0, suggesting that, you know, this high consistency that these resin ducts are good correlates for of resistance to bark beetles. So phloem ducts, again, we just don't really know hardly anything about the phloem duct system in trees. What little data is out there just in the past few years shows it seems to be pointing to that it's mostly related to a constitutive defense instead of induced. But our study set out to try to sample this and investigate that more. And then we also wanted to look at not just constitutive defenses, so the baseline defenses that pines always have, but what about how well do they induce? So we know pines can increase their resin flow. This was a study where we mechanically wounded the trees and tracked resin flow over time. So the unwounded trees, or the control trees, are in white bar, and gray are the wounded trees. And at about 28 days after the wounding is we start to see an increase in resin flow. And we know that increase in resin flow also can relate to changes in induced chemistry. So this is from the 2017 study where this is just looking at Pinus aristada and Pinus flexilis. And on the left are the constitutive defenses. So Pinus aristada has higher constitutive defenses than limber pine and much more inducibility. So if you look on the right graph and notice that the y-axis is has a much higher scale compared to the left graph. And so the existing resin ducts are ramping up resin flow and changing the actual composition of the resin. This was shown also in the other species from the 2019 studies, where we found foxtail had the highest inducibility of compared to flexilis and longaeva. So taking that all together, you know, we still hadn't looked at how these resin ducts related to resin chemistry. So we wanted to focus on this, but also bring together all the data from these 3 studies and just kind of try to put everything together as much as possible. So that brings me to the results. So this is just a table of the basic tree characteristics, just to give you a sense of the sample sizes and just overall kind of characteristics of the trees we measured. You'll see that the number of trees for basal area increment or tree growth is a lot smaller, and that's because we don't have growth cores for the 2019 study. But overall, the DBH was smaller for Pinus aristada, and highest for for Pilo and Piba, or foxtail or great Great Basin bristlecone pine. Foxtail had the highest growth rate and also the highest or thickest phloem. So we investigated phloem a little further, again, because phloem is where the beetles are spending their the majority of their life. And we know that phloem thickness is positively related to bark beetle fitness, and bark beetles do better with prefer thicker phloem. So once we accounted for, we realized we needed to account for basal area increment or growth rate to look at phloem thickness. And after accounting for BAI, the three sister species of the two bristlecones and foxtail all have the same phloem and thicker phloem compared to limber pine for a given growth rate. Okay, so moving to xylem, ducts, and growth, the graphs I have are all set up the same way with the 4 species on the x-axis, all in the same order, and then the different resin duct metrics on the y-axis. And these are 15-year averages, so the 15 years prior to any induction. And what you can see here is that foxtail and Great Basin bristlecone pine, Pilo and Piba, have the largest resin ducts, with Piba having the highest. Piba also has the highest area in resin ducts. And then looking at the other ducts metrics, we don't see differences in resin production by species. Relative area, which is a standardized metric, is highest in Pilo and also in for duct density. And then this is just the same data as the table showing that Piba had the highest growth rate, followed by Piffle for our trees. So now moving on to phloem ducts, this gets a little more complicated because now we have both constitutive, which is shown in the orange-ish bars, salmon color bars, and then, so that's with, and the blue bars, so that's the 0 times 0. And then F are the trees that were inoculated with a fungi, and M are the trees that were mechanically wounded. So what you can see here with phloem duct size is that the bristlecone pines and foxtail pines all have higher or larger resin ducts compared to Pinus flexilis, but there's really no indication of inducibility within 30 days. And that was the same for phloem relative duct area and also for duct density. And in hindsight, it's perhaps not surprising that we didn't find any differences within 30 days. Likely, it might just take longer to form the actual ducts compared to changing flow or chemistry. So this might have been something we needed to look at for longer. Okay, so now moving on to the xylem duct and growth correlations. I know this is a lot, so I'll try to step people through it. But these are all set up the same way, where each block are the correlations for each of the 4 species, with the resin and the resin duct metrics and BAI and phloem thickness. And so the warm red colors are positive correlations, and the purple cool colors are negative correlations. And so to kind of point out some of the general patterns that we found, xylem duct size, total duct area, and production were all positively related with each other for the species. Relative duct area was positively related with duct density. And looking at our unstandardized metrics, which are size, total duct area, and production, were negatively related with the standardized metric of density. And so a lot of this has been shown in other pine species, but we haven't investigated it for these four. And then looking at how xylem duct relates to growth and phloem thickness, we found that faster growing trees, so that higher BAI and thicker phloem, were positively correlated with total duct area and production, and then somewhat with size. Size was like, I didn't mention, I should have, but if only the significant correlations have the numbers recorded. So if the box is empty, that it shows the direction of the correlation, but it was not statistically significant. And so because that these faster growing trees had higher total duct area and production and size, it suggests there's little trade-off between these xylem duct defenses and growth. Okay, so looking at phloem and xylem duct correlations, unfortunately, we could only do this for 2 species because we didn't have the growth cores for the trees that we had the phloem samples for. But from those, we found that for both PIR and piffle, that phloem duct size and phloem relative duct density were correlated with the resin duct different resin duct metrics, and growth. Again, these negative correlations are very common with the standardized duct metrics, and then positive correlations for the unstandardized metrics. And we found that phloem duct density was just not correlated with any other phloem duct metric, xylem duct metric, or growth or phloem thickness. Okay, so moving on, this is kind of my last data slide about how the xylem ducts and phloem ducts relate to chemistry. So here we have the MT constitutive is the total monoterpenes at the constitutive level, and total monoterpenes on day 30 with a fungi inoculation, and then mechanical inoculation, and then the ST stands for sesquiterpenes. So just looking first at constitutive defenses, we found that limber pine seems to have the opposite correlations with the unstandardized duct metrics, that they have having negative correlations, whereas PIR, PIBA, and PILO all had positive correlations, although it was much only significant primarily in PIR. And then looking at phloem relative duct area, they had consistently positive relationships with the constitutive monoterpene levels. Okay, so now focusing on induced chemistry. Again, that the limber pine and Rocky Mountain bristlecone pine seem to have largely opposite patterns and between whereas the limber pine has a lot more sesquiterpenes than PIR, and you see positive correlations with induced sesquiterpene levels for both treatments in the xylem ducts, and you don't really see any correlations at all with sesquiterpenes for induced sesquiterpenes for PIR. For the monoterpenes, we see a much stronger response for the fungi treatment compared to the mechanical treatment for PIR. And it seems to be the opposite with piffle. And then we don't have, and then let's see, for PIBA and PILO, we don't have a lot of the data to test the xylem ducts, but for phloem ducts, we see that this the sesquiterpenes for mechanical and monoterpenes for mechanical are both positively related to phloem duct density. But looking at the difference between constitutive and induced, we don't see much to suggest any trade-offs occurred between constitutive and induced chemistry and ducts. Okay, so in conclusion, a lot of different data is showing that these defense traits and correlations are generally species specific. We didn't find many trade-offs or evidence of trade-offs for growth and defense or constitutive and induced defenses for phloem ducts or resin duct chemistry. And that taken these results, or given these results and a lot of past studies, it seems to suggest that pine resistance can emerge from multiple different defense trait combinations, and that these trees have learned to utilize their defenses in different ways, on a species specific basis. And so that's all I have. And I just want to, again, acknowledge my co-authors, especially Barbara Benz, who's kind of organized and kind of these studies were her brainchild, and Justin Runyon. And also thank Forest Health Monitoring for their funding of 2 evaluation monitoring projects. And with that, I can take any questions.
Hey, awesome. Thank you so much for your talk, Sharon. So yeah, I'd like to open the opportunity. I'm Karina, by the way. I'm going to manage the Q&A. So now is your time to ask your questions. And if you can just throw them in the chat box, that would be great. We have, let's see, about 15 minutes or so, a little over 15 minutes. So yeah, go ahead and put your questions in there. We do have kind of a specific question already from John Van Gundy. And he was wondering if you knew, Sharon, who's replacing Chris Stalling on the High5 whitebark pine database for the National Whitebark Pine Recovery Plan. And I am going to have Julie chime in to kind of tell the audience what that is exactly. You're welcome to also.
Yeah. Sharon, do you know anything about the High5 database, or you can explain what it is if you want, and if you have any information on that, or I can give background if you don't.
I mean, I know about it, obviously, but I mean, you can talk. I don't know about your, I guess, what plans you
basically it's a database.
Yeah. Okay. You want me to explain? Yeah. So it's a database.
Yes.
That I think started
it looks like
with, I think Bob Keene and others initiated it with looking at different, it's basically an Excel spreadsheet of reporting all the different responses for whitebark pine studies. But its main, Willis. So, and don't you have a link to it on your website, right? On the Whitebark Pine website or Whitebark Pine Ecosystem Foundation? Or not?
We do not have a link to it right now. And
okay.
It looks like my internet's unstable, so I'm not sure if you guys can hear me very well. Sorry about that.
We can hear you. There was a little bit of lag, so it was hard to, yeah. think people were talking about.
I guess the short answer is Chris retires this month. And well, I guess we just, I guess we should, I guess we need to discuss that, but I don't really know who might maintain it right now.
And we're also talking, I've talked to Chris about it a bit, and we're working together with the folks at the Fire Lab and the agencies to kind of figure out what the best way to move forward. There is some data that's been updated recently. I think John Van Gundy and your group have submitted data, but then there's a lot of other data that has not been submitted or included recently. So it definitely, if we're going to maintain it, it needs to be updated significantly. Some of the data in there is really old, from the 60s and 70s, and some of it I think came from literature searches. So there's a lot of cool stuff in there, but it definitely needs to be gone through and updated with more recent data, and a way for people to start submitting data again if we're going to continue to use it.
Cool. Yeah, it sounds like a great resource. I'm always curious, like, how to find the, most up-to-date distribution for these different species, and I'm sure I'm not alone there. Okay, great. Yeah, and looks like we do have a comment from Diana Tomback, who says that it was initiated by WLIS, by Forest Health Protection, and then has been maintained by WPF for a while, and then it moved to the Rocky Mountain Research Station. So it's a little bit more info. Okay, let's see. Our next question is from Laurel Sindewald, and she asks, in reference to what you found for limber pine, do you know if there are trade-offs between drought tolerance traits and growth, or drought tolerance and defense?
Yeah, that's a good question. So I guess I know more about how drought can affect the resin duct defenses and chemistry, but we haven't looked, I don't think there's been any studies looking at like hydraulic safety margins and xylem structure that would lead to kind of at least xylem hydraulic traits, and they're related to drought and defense. No, it was that. I don't know if that answers the question, but so
yeah, I think that's it. Yeah.
Yeah, not that I know of.
Not that you know of. Yeah.
Yeah.
All right, let's see. Yeah, so you talked about 3 different studies, and one question is, what would you have done differently if you could do it all again? The big question for researchers everywhere. What have you learned?
Yeah, I guess because it's made it, at least for this part of the study, trying to wrap up with kind of publish all the data from the 3 studies with the resin ducts. It's in hindsight, not measuring the not coring the trees first of all, not tagging the trees at the beginning. I would say, if you can, always tag your trees, because you never know, you might want to go back to them. So having good monumentation would have helped us measure those exact same be able to measure the same trees. But then really trying to, we made the decisions that we did based on funding levels and just the complexity of getting to all these sites, that they're hard to get to. So it takes a lot of travel time, and then to return to them, it takes a lot of time and money. So we decided not to measure, core the trees to measure resin duct data, and for that 2019 study. And that was really, I guess that's the big hole that kind of drops us down to 2 species comparisons instead of 4 for some of that. So, so yeah, in hindsight, it would have been really great to we would have had more consistent data throughout all 3 studies.
Got it. Yeah. John Van Gundy brought up the idea of using GPS coordinates, which I think is something that we tend to do nowadays. But I do agree that having the trees actually tagged is great, because then if the tree is no longer there, you can kind of know for sure. Whereas, yeah, I think with GPS.
And often, depending on your I mean, you can get a high accuracy GPS and they're improving, but it might be great. It's great for a plot, but finding the exact tree can be, you often need something more than just a GPS coordinate.
Yeah, having that confirmation is just nice for peace of mind, I would say. Yeah. Okay, great. Let's see. Melissa Jenkins asked, can you provide any recommendations for managers based on your findings? How can we apply this?
Yeah, I know a lot of these results are very, it's a lot of basic research that we just trying to understand these defenses. Kind of what has come out of the sampling and these sites and further research that especially Barbara has done is, we have found that Great Basin bristlecone and foxtail are large. I mean, they're not great hosts for mountain pine beetle, but there are, for Barbara has found that for Great Basin bristlecone pine, it's mountain pine beetle and ips will attack them under extreme drought conditions, and when they're co-occurring with another host like limber pine. So those are areas that we might expect some mortality to begin to increase if managers have those kind of co-occurring areas and their land base, especially under extreme drought conditions. And so, if there's especially important populations, you would trying to limit the host species would reduce the pine. Basically, the mountain pine beetle does not really want to attack Great Basin bristlecone pine, but it doesn't really the beetles don't reproduce in the tree. It acts as a population sink, but given that some of these populations include hundreds or thousands-year-old species, we don't want to lose those genetic resources. So I feel like I guess managers should know that those areas are going to be more vulnerable than like the highest, the populations where they're not growing with other host species.
Got it. Yeah, that's interesting. I guess that kind of leads me to the question, do we know why Great Basin bristlecone pine is so resistant to mountain pine beetle? Like, what's special about it?
We don't know exactly. I mean, it seems to be related to the monoterpenes, are not just monoterpenes, the terpene, basically resin chemistry, and it seems to be, it's probably related to the volatiles in the resin, to where the trees just are not attracted to the beetles. The beetles are not attracted to the trees at all. And so, but we don't really have a sense of it, like exactly what that mix of terpenes, is that makes it so much more resistant than any other pines that we know of.
Got it. Yeah. Diana Tomback was chiming in here that the wood does seem very dense. Could that have anything to do with tolerance also?
Yes, in the first study, the 2014 study, we did look at wood density, and yeah, you're right, Diana, that it is very dense. I'm trying to remember, it's really, I mean, it was much denser than and so, you know, we're still, you know, we thought about these things a lot, and are these, we call them defense traits because we know they're related to resisting mountain pine beetle. So that increases the tree's fitness in the presence of beetles. But these same traits also probably help confer their longevity. So resin, you know, especially on the highest elevation species, when super old, like you get the strip kill, and these trees are able to stand for centuries, kind of half dead looking, you know, whereas a lot of other species are lower elevation, there would be more fungi, and the trees just would mechanically fall over or rot. And so this resin, the amount of resin that's produced, and that's right because of all their high wood density and their high resin duct system, allow that wood, as it dies and is strip killed, to kind of be preserved for much longer, so that it can it's not necessarily, these same traits aren't necessarily only used for pine defenses. They can also have other benefits to the tree.
Got it. Awesome. Let's see, we've got a question from Richard Sniesko. I hope I'm pronouncing that okay. Any thought of correlation between mountain pine beetle resistance and white pine blister rust resistance? Is there, yeah, like an interaction there?
Yeah, I don't know a whole lot about that. I feel like it comes up, and I know Anna Shuttle looked at xylem resin ducts, I think, and it was in limber pine, and related that to white bark pine blister rust resistance, and didn't find any correlations. And I think that's the only study I'm aware of. But I don't know, that's a good question, to see if there's other research out there.
We are wrapping up pretty soon. So, yeah, Laurel's like Sinesco. I actually have heard that name, but it is funny how it's written in the chat. So that threw me off. Anyways. If anybody has any other last minute questions, now is the moment. But I always like to ask Sharon, you know, what's next for you? What is your kind of like next research goal at this point?
That's a tricky question these days as a federal employee.
I understand. Yeah,
So
yeah, maybe
right
you
now,
don't want to look
I'm
too much
hoping
into
I
the
can complete
future.
a field season and continue, yeah, on with some of my studies. So, but
Yeah.
yeah, we'll see.
Okay. Great. Well, yeah, I'll go ahead and close up this webinar then. And yeah, we just like to say on behalf of the White Bark Pine Ecosystem Foundation and the organizing team, we just want to thank you again, Sharon, for participating. It is so great to have experts like yourself weigh in on these topics. And, you know, like myself, I see your name all over the place in the literature, and it's always great to put a face to the name and kind of get it from the horse's mouth. So we really appreciate it. Our next webinar is on May 18th. And we have Vlad Kovalenko and Enzo Martelli, who you all know, presenting their work, Understanding the Effect of Restoration on the Primary Seed Disperser of Whitebark Pine on the Sawtooth National Forest. So we are really excited for that. And thanks again for everyone for attending, and stay tuned for our upcoming installments. Have a great day, everybody.
Hi, thank you.